This is Sanjita and I'm going to discuss about one of the tasks in for my biocomputing HackBio internship for the stage 2. In this stage, actually, I'm going to analyze the RNA-seq data for to identify the differential gene expression and their function. So these data actually pre-processed data, and this gene expression data came from by using the RNA-seq experiment. So in this data contain actually a diseased cell line with the treated ones. So we can first see the data in here, like the gene name, log2 fold change and p-values and p-adjusted values. So as I save the data in my local computer, so I can download the data from here by using the read.csv function. So then I want to visualize this data by using the Volcano plot. And so in part 2, we are going to see the data Volcano plot in data visualization part. So this actually the Volcano plot is one of the scatter plot which is widely used in differential gene expression analysis to visualize the significant change among different genes. And this plot is actually really helpful, especially like when we have a very big data of RNA-seq data, and this data actually help to visualize the significant change among the data. So for this, first we have to set threshold to see the log fold change. Like log fold change 1 means the gene fold change increase 2 times, and the p value actually shows like the significant p value changed. But in here we use the adjusted p values because like it will avoid the false result. If we use the raw p values, it will from a big pool of data or the genes, sometimes it shows the false negative result. That's why we will use here the adjusted values. So first we want to add a new column which will show the up and down regulated gene. So we'll here use the if else function. So this is actually the conditional function which will show like if the condition met, then it will be the upregulated, if not it will be the downregulated. So our log2 fold change will be the higher for log fold change, the cutoff values, then it will be the upregulated. If it's lower, then it will be the downregulated gene. And to visualize these up and down regulated genes, we'll plot the Volcano plot in here. First we assign the colors. We will use the same function if else. So the upregulated gene will show the red, and the downregulated will show the blue, and the gray one actually didn't pass any of this condition. So in y-axis actually we'll use the minus log 10 p adjusted values to plot this data. So in x-axis it will come to the log 2 fold change, and in y-axis it will show the log 10 p values. So this will show the 16 means like the solid points, and we'll assign the color in here. So this shows the x and y label. Main means it will shows the title of the graph, and C6 means like 1 point. It will show actually the point size, like what is the size we want actually. And x limit means like we want to make it the x-axis in symmetrical way, so we put in these positive and negative values. So then if we want to plot in here, so I already plotted in here. It shows clearly the red one is the upregulated. So from this data we find like 1 upregulated and 4 downregulated genes. So there are some couple of important of this Volcano plot actually. When we have a large RNA-seq data, so this kind of plot can make it easier to visualize like which genes are significantly expressed and which are significantly decrease their expression. So it's really easy to visualize from a big data. And normally the genes which is highly expressed based on their cutoff value shows easily in this plot. So After that, like I want to subset the genes to find separately the genes up and down regulated genes. So in here we try to filter from the data based on the cutoff values up and down regulated genes, so we can print the result. And in here it shows down and up regulated genes. We can save this result as a CSV file or any file if you want, by using the write.csv function. So from here, like after finding this gene, I want to see their function, so I can just copy one gene in from here, and I'm going to use the GeneCard functions. So for example, like if we go to the GeneCards database and explore a gene under this section, like if we just paste the gene name, then you will see like all of the information about this gene. Like in here, if you want to search like their function, like their disorder pathways, and their drugs, related drugs if they have, I want to see their functions. So it shows like this gene is maybe responsible for anchoring smooth muscle cells to elastic fibers, or maybe it involved not only in the formation of elastic fiber, the process of this vessel assembly, or have the capacity to sell adhesive. So in here we can see their molecular function. In addition, we can have a look their pathways, so maybe it could be connected with some specific pathways which maybe we can use as a therapeutics or drug target. Again, we can have a look the drugs, maybe they have some responsible drugs. So from this database, also from if we go further, we'll see like there are some related journals. So from here we can see like how previously studied this gene and what they have found there. We can just manually search all of the gene up and down regulated gene in here, and we can find their related informations. So in this way, like we can plot a volcano plot by to see the differentially expressed gene and their related functions and their drugs and other informations. Yeah, so thank you.